So indeed, my talk will be about charting brain connections with diffusion MRI. This is me. Okay? I'm the physicist. This is what I do for a living, looking at equations, trying to solve them, implement them in my daily practice. Just a quick question. Your background, so like, who's a medical doctor or in training? Just, can I raise some hands? Medical doctors? Yeah, just a few, okay. Physicists? No physicists? Engineers? Nobody's perfect, don't worry. All right. Anyway, this will be my only slide with equations, so don't worry about it. And in matter of fact, I actually work at a medical center, so one of the largest medical centers in the Netherlands. And it's very interdisciplinary. I work at the faculty of medicine, and within the medicine faculty, we have many, many different departments. And in this department, I'm working on diffusion MRI. And I'm working very closely with clinicians, right? So they do their clinical research, and we have a very nice feedback. So on the one hand, we develop new tools, and on the other hand, we've got the clinicians that have, let's say, challenges. We don't call it problems, right? Challenges. And we try to facilitate them and help them out. Quickly gonna switch. Hopefully that'll be better. Okay. So, okay, then I have to turn this off, I guess.
Just use the normal mic. This one.
Okay. And so the nice thing about this feedback loop is that we've got our identity within the facility here. And so a few years ago, I set up a research lab called the Prof Lab, where we focus on processing, analyzing, and visualizing diffusion MRI data, and specifically for looking at micro tissue. And so for many clinicians, diffusion MRI, they often talk about confusion MRI because it's quite a difficult topic. And so for those few medical doctors in the audience, I will try to explain diffusion MRI in a very simple way. And for the engineers, I'll quickly start with showing what a brain is. Okay? So this is how a brain looks like. If you cut it open, you can clearly see different tissue types, and I want you to focus on the main ones, which are gray matter, the cortex, and the white matter. And for the remainder of this talk, I would like you to focus on the white matter. Why? Because it basically consists of this fibrous tissue. Think of it like spaghettis, right? Think of your white matter of your brain as spaghettis connecting different brain regions. And this is how it would look like, if you've got this straight fibrous tissue. MRI, I hope we're all familiar with MRI. If not, here's my 3 slides on MRI. Well, you've got this big machine, you lie in, and then you can scan the internals of your body, right? So in this case, looking at these images, we're looking at the relaxation properties of induced tissue magnetization. And the cool thing is that, already 5 Nobel laureates have been involved in this work. So, pretty important stuff. What about diffusion? Okay, so diffusion. Well, think of diffusion as putting a drop of wine in a glass of water. And if we wait long enough, then all these wine molecules, they will spread out. They will diffuse in the glass. And you end up with this kind of homogeneous looking, glass of water with this drop of wine. Microscopically, if you look at it from a particle point of view, if you, for instance, consider these red particles and the green particles, if you remove the white barrier, you can appreciate that this is driven by thermal energy, by random motion. Okay, so these molecules, they bombard into each other and they spread. This is also called Brownian motion because it was first discovered by Robert Brown. And as you can see, he was very happy with his discovery there. Happy chap. An important concept that I want you to understand about diffusion is the difference between isotropic and anisotropic diffusion. That's the typical reaction most students get when they hear about anisotropic diffusion. Sounds very difficult, but it's just fancy to say something about the type of diffusion. Okay? So let's start with isotropic diffusion. What is that? I'm going to show the same animation again, but then with the red particles colored in the middle. Okay, so let's start the animation, and now we can appreciate that the spread of these red particles is the same in each orientation. The rate at which they spread, up, down, left, right, is all the same. Okay? By contrast, well, actually, you can also visualize this with a circle or in 3 dimensions with a sphere. So this is how you can quantify or characterize this geometrically. And by contrast, anisotropic diffusion, which is just the opposite of isotropic diffusion, well, consider, for instance, some barriers. These could be cell membranes or neural fiber, axons, things like that. Then it's not hard to imagine that the diffusion will be like this. So here, you can clearly appreciate that the diffusion is more pronounced horizontally than vertically. Okay, so this is what we call anisotropic diffusion. And we can visualize this with an ellipse in 2 dimensions or in 3 dimensions with an ellipsoid. Okay? So that's diffusion in a nutshell. And what I want you to remember is we've got something which is called anisotropic diffusion, where the spread of these molecules or these particles is pronounced along a specific direction. So what about diffusion combining with the brain? Well, we know the brain, especially the white matter, has this very organized tissue. Well, then it's not hard to imagine that if you have water molecules, because our brain, 80% is just water molecules, just diffusing through our brain, then all these water molecules, which we can image with MRI, and I'll come back to that later, these molecules, they also spread, and they kind of feel, they probe the microstructure. So you've got these particles moving in our brain, and they're following the orientation of the underlying microstructural pathways. And that also induces anisotropic diffusion. Okay? So the cool stuff is now doing something with MRI to try to capture this diffusion process, because if we can quantify the diffusion process at every location in the brain, then that diffusion profile says something about the underlying tissue architecture. If you look at conventional MRI, we have a very high resolution nowadays, a sub-millimeter resolution. And if we look at the centrum semiovale, for instance, we can clearly see, the difference between white matter and gray matter. But within the white matter, we're still flying blind about what fiber pathway are we looking at. Like in the centrum semiovale, this area over here, we've got projection fibers, association fibers, commissural fibers. They're all running, in different orientations. And it's that orientation that we could not capture with, let's say, conventional MRI techniques. Okay, and this is where diffusion comes into play. And I will skip the entire part about the acquisition. It took me, 2 years to really understand how it worked to begin with. But what it basically does is, with conventional MRI, we just take one image, right? And we get this grayscale image. So every pixel or voxel has just a gray value. What we're doing with diffusion MRI, and I hope you can appreciate that this is actually an animation, you can see the contrast changing in the image. What we're basically doing is that we're sensitizing our diffusion, the rate of diffusion and the orientation of diffusion at the same time while acquiring an image. So we acquire an image and we look at the specific orientation, say like, how much diffusion is there in this direction? And then we acquire another image and we just say like, okay, how much diffusion was there in this direction? And by probing all these different orientations, we can get a nice 3-dimensional profile of our diffusion at every location in our data set. And so it's kind of like this, right? So you look at this orientation, you see that, yes, there's diffusion. And then you rotate it, no diffusion in that orientation, right? And so you probe everything, and by doing the full characterization in 3D, you get an idea of the object. right? So in this case, if I were to look at only this direction, I get a yes. If I only look at that direction, it's a no. But you want to have the full description. So that's one of the current challenges as well, is trying to optimize the acquisition so it becomes quicker. Because nowadays, an acquisition of a three-dimensional data set is still in the order of 5 to 10 minutes, whereas in a conventional T1 weighted image, for instance, just half a minute or something like that. Okay, so every voxel of our data set is now a series of scalars. Like, for instance, with functional MRI, if you're familiar with the bold signal, you acquire many, many images, and those reflect, let's say, your activity in the brain, from which you can then compute which brain regions were active for which task, etc. Well, with diffusion MRI, you got this array of values, and they represent the amount of diffusion along a specific orientation. And the idea now is to try to quantify this. And this was proven to be very difficult. And Peter Basser in the early 90s, he came up with a framework, a diffusion tensor framework. So he used a mathematical formalism to basically quantify this diffusion in a rotationally invariant way. Because you can imagine if I lie in the scanner differently, well, the relative orientation of a diffusion orientation becomes different if I rotate my head. So we had to come up with this rotationally invariant framework, and with using a simple PCA analysis, you can figure out what are my diffusivities along these main axes. And so for every voxel, you now end up with a diffusion ellipsoid or a diffusion tensor. And so if you look at it medially from, at the right hemisphere, you get an image which looks like this. And I've got 2 young kids, and a while ago, I went to pick up my kids from school, and the teacher, she comes up to me and she said oh, yeah, I didn't know you worked in a candy factory. I was like, what? Yeah, well, apparently I showed this image to my kids, and I didn't properly explain to my kids what Daddy does for a living. So yeah, now they think I work in a candy factory. At least I'm very popular at school now, right? So people asking for candy all the time. Anyway, so as you can imagine, looking at an image like this is not convenient, right? How can we get a better idea of the underlying architecture? So instead of showing an image map like this, again, this is now an axial slice through the brain, where every pixel or voxel is showing this ellipsoid. It's not very convenient. First of all, the color in this case is defined by the orientation of the ellipsoid. So if you look at the main axis of the ellipsoid, you can just think of it, if that is an orthonormal vector, you can take the absolute value, you get 3 values, and you can map it to RGB values, right? And then you get the colors here. So red means, for instance, 1, 0, 0 is left, right. So everything shown in red here is some ellipsoid pointing left, right. Everything in green, 0, 1, 0, is pointing up, down. So everything, pointing up, down is in green. So it's basically enhancing your visual cues to look at this image. So why not map that property directly to a map? Okay, we can do that. And simultaneously, we can also quantify the intensity of that map with the eccentricity of the ellipsoid. So if the eccentricity is very high, meaning like a cigar-shaped ellipsoid, we'll give it a value approximately 1. And if it's very spherical, we'll give it a value of 0. Okay, and then you end up with a map like this. So this is exactly the same information, but then put in a map. And this is what medical doctors obviously prefer to look at, right? And so what does it mean? Well, again, everything which is dark has approximately a 0 value. 0 means spherical. Spherical means there's no preferred orientation of my diffusion. So these molecules, they just, wander around, left, right, up, down. By the way, I'm walking like this. This Brownian motion is also called a drunken man's motion. So if tonight at the gala it becomes a fun party, I'll be walking like this as well. So this is also the Brownian motion. Okay. Anyway, a side note. So if I look at this map here, everything which is dark, like for instance this area over here, well, there's no preferred orientation. If my water molecules can diffuse freely, what's my underlying tissue? CSF. It's just water, right? It's the cerebral fluid in which molecules can move freely. There's no preferred orientation. So what about the bright areas? Well, everything which is bright has a high eccentricity, cigar-shaped, clear preference in my orientation of my diffusing molecules. Well, that means that there's a clear underlying microstructural architecture going in a specific orientation, and focus on this structure. It really pops out here, right? Well, this is part of the corpus callosum, connecting left and right hemispheres. So that's what you see over here in red, left, right. And here it's actually arcing a bit to the back, so it becomes more greenish. And obviously all the other colors are just intermediate orientations. Okay? So, in this case, what's the benefit? Well, if you look at the T1, again, very high resolution, very nice differentiation between white matter and gray matter. Diffusion information, you've got high specificity about the orientation of the underlying tissue. And obviously, if you combine both, you get something like this. And if you, again, focus on this middle part here, you can clearly see pathways going up, down, which is 0, 0, 1, up, down, which is blue. So blue is pathways going up, down. So these are probably the motor pathways, corticospinal pathways. In green, front, back. These are association pathways. And then in red, you can see pathways going left, right. This could be, for instance, lateral projections of the corpus callosum. So we become a lot more specific. And just to showcase this to the medical students in the audience, you've probably seen an MRI image like this before. And if I ask you, anybody of you becoming like a radiologist or in, seems like you look very junior yet, so no specialization there, but no radiologist. Anyway, I asked our team of radiologists, and we got 25 in Utrecht. And I asked them, do you spot anything abnormal here? None of them said, well, there's something abnormal. So for all of them, it looked normal. Okay? But then I showed the corresponding diffusion. Let me just zoom in. Well, what do we see here? We see a green patch in the middle of a red patch. Remember, red means left, right, and that structure in the middle of the brain connecting left and right hemispheres, I mentioned before, is the corpus callosum. But there's a green patch in the middle. What's going on here? And what that green patch basically represents is a hyperplasticity. So in this case, the subject had pathways going front, back in the middle of the corpus callosum. Okay? What does it mean? We don't know. But what it does say is that our brain is more versatile, more complex than we thought it was. The cool thing about having this specificity is to look at rare conditions like callosal agenesis, right? People who don't have a corpus callosum. Like Kim Peek, you may have seen the movie Rain Man, for the older people in the audience, with Dustin Hoffman and Tom Cruise. This person is actually based, so the actor, Dustin Hoffman, he basically, his character was based on Kim Peek, who missed a corpus callosum. But he couldn't button his shirt because you need both hands working together to button your shirt. But he could remember everything he saw, which was also kind of a curse, I can imagine. But he opened the telephone book. In the olden days, we had a telephone book. And he could remember every phone number from anybody in the States. And he could play the piano in a very weird way because he could use his hands independently. And so he it's kind of amazing. So with this technology, you could also then look at these Probst bundles. So people who don't have a corpus callosum, they have other pathways. So their corpus callosum basically reshapes into another structure. And this is something you couldn't investigate or study with conventional T1 weighted MRI, but you can with diffusion MRI. So increase in specificity about the orientation of the white matter pathways. So one of the main applications of this diffusion MRI is obviously to actually quantify these pathways. Because what our brain already does is kind of looking at these ellipsoids, and we're connecting these ellipsoids. Okay? And this is called tractography. So we're basically looking at the main orientation of this ellipsoid, and then we're connecting these discrete elements. And in a way, it's similar like this, right? If I ask my kids, just connect these little lines, they can do this easily, right? They just connect these discrete elements. And, in science, we can do the same. We can do vector field interpolations, Runge-Kutta interpolation, and get something similar. So, to do that, you have starting points in your data, and then you just connect the discrete elements. And by doing so, we can now virtually, non-invasively, in vivo, reconstruct white matter pathways in the brain. Proud to be a nerd. Anyway, so for most people, this was just a wake-up call, some of them falling asleep, so that's why I put in the music. But this is how a tractography starting point would look at when it's traversing through this diffusion tensor space, right? And at each position in space, you would be looking, okay, what's my next position? And you follow it. Similar like in fluid dynamics, following the current, the stream of a river, right? And we do exactly the same with these data. And in practice, we don't just sample a few points, we sample the entire data set very densely. And then you end up with this kind of spaghetti-like structure, which we call a whole brain tractography data set. And I once had a junior radiologist came up to me and he said, "Oh, Alex, I didn't know axons were that thick." And so I was like, "Oh, these are not axons, right?" Oh, okay. So I kind of joked like, "What about the color?" And he said he first wanted to say something like, "Oh, I've never seen a patient with these colors in their brain." Well, duh. This is all CGI, right? I mean, the pathways that we reconstruct, they don't have an actual thickness, right? These are just space curves, right? Just connecting points in space. And just to drive home my point here, what we're looking at are reconstructed pathways from a diffusion process. So we're not directly looking at pathways. Okay, admittedly, if you look at some structure like this connecting the orbitofrontal cortex with the temporal pole in a hook-shaped manner, well, the uncinate fasciculus springs to mind. So it looks like it's doing something properly, right? But the thickness, again, is just to convey the architectural configuration more clearly. If I were to show these space curves, like on the left, with no shading, no thickness, well, if you look at the right, it's more clearly depicted. And just to prove the point, here are two uncinate fasciculi. One is the left, and one is the right. You can't tell by looking at it like this, but if you look at it like this, you can clearly see that the left image is the right uncinate fasciculus, with radiological convention. But the cool thing now is that we can reconstruct pathways in vivo. So think of it as like a virtual dissection or in vivo dissection. And as you can appreciate, if you have some knowledge about neuroanatomy, you can see, for instance, in blue, part of the fornix bundles. Again, the uncinate fasciculus in green, and optic radiation, optic tract in red and yellow. Okay, so many of these pathways that we're familiar with, we can reconstruct in vivo now. And this is just visual, right? We can also quantify properties underlying the tissue microstructure. Again, think, for instance, of the eccentricity of the ellipsoid. Imagine that you have a pathology, right? What happens to your brain? Well, you've got microstructural degradation, right? Let's call it the integrity of the white matter being compromised. So tissue filaments, they break down. Well, the myelin maybe goes away. What happens to the diffusion process? Well, the diffusion will change, right? So for instance, the anisotropic behavior will become different. This is quantified with something called fractional anisotropy. It basically states to what extent is my ellipsoid cigar-shaped. Well, if it's very cigar-shaped, we think of it as being healthy, right? Very, nice myelin, axonal filaments all intact. But if that degrades due to pathology, then this anisotropy goes down. Also, the opposite is observed when you're just developing as a young child into development. You can clearly see here, for instance, in the corticospinal tract, that this eccentricity, this fractional anisotropy, or in short, FA, it increases with age. This is kind of what you expect, right? A child develops, their motor skills get better, so their motor tract gets more coherent, think of it like that. And so we can quantify properties of the underlying white matter. And obviously, there's always the question but you make assumptions, the data is not perfect, there's noise, the model assumptions, there's some parameter settings involved when doing tractography, like when you take a little step, there's this angular deviation. How much angular deviation do you allow to progress? But if you look at these images, this is histology, this is in vivo, we're doing a good job looking at, let's say, the highway nature of these pathways. And, you could say, if it looks like a duck, quacks like a duck, flies like a duck, and maybe even tastes like a duck, it's probably a duck, right? So we're on the right track, I would say. Another application is neurosurgery. Well, if you look at the diffusion map, you can be a lot more specific towards neurosurgical navigation. From what entry point should we do the surgery? You want to minimize collateral damage. You want to avoid the eloquent fiber pathways, so you're not, doing too much damage to the subject. And we can now even use this technology to look at gray matter, because gray matter is also very structured, although a little bit more complicated, because you've got all these layers, right? And so our resolution with diffusion MRI is becoming high enough to also quantify properties within the gray matter. Here's just a quick other application. So we were talking to some biologists, and they were investigating this brain of, anybody any idea what kind of brain this is? No? Well, it's actually a starling brain. And you may wonder, a starling brain? Well, a starling is actually a songbird, and they can mimic sound, insanely well, right? There have even been reports of starlings mimicking chainsaws and everything, I can't. Anyway. So why study the songbird? Well, it's a very nice model for speech. Okay, so in this case, they were looking at 9 male starlings, and they were wondering, are there any differences in their brain architecture between spring and summer? Because in spring, they're all horny, they want to mate, and in summer, they've done their thing, they're all laid back, right? So does that also show in their architecture, right? So a lot of, whispering and all the singing in the spring versus summer, nothing at all. Very long story short, yes, we could find through some voxel-wise analysis regions corresponding to nuclei that are related to song and speech that showed significant differences. And the same using tractography. So it seemed like in spring, we had stronger connections, more pathways connecting these song nuclei. So that was very exciting especially for the biologists. I was just a facilitator with the technology, but I'm happy to be involved, obviously. Okay, so life of a diffusion MRI scientist must be awesome, right? I'm going to show a confession by a famous diffusion MRI scientist.
Yeah. Yeah, so I think this was back in 2004, yeah. I was stood outside of the operating theatre, and the neurosurgeon came out to me and told me he was thinking of making a cut in the patient's brain, and he wasn't sure whether to go near the corpus callosum or near the corticospinal pathway. So I used my software and I said to him, look, I can see the corpus callosum, it's all lovely, but I cannot see the corticospinal pathway. So, on balance, I would cut near the corticospinal pathway. Anyway, as a result, the patient died. Yeah. I suppose, looking back now, you could say the tractography was a bit of a letdown, particularly from the patient's point of view. So, what did I do wrong?
So obviously, it's kind of funny, but unfortunately, it is not funny because there's some reality attached to this when technology is being transferred to the clinic, right? We want to make sure that our technology is correct. Well, talking about model assumptions, I was referring to this diffusion tensor, this ellipsoidal framework to quantify the diffusion profile, right? But this kind of assumption may not hold if we think that the brain is a lot more complex than just coherent spaghettis running nicely parallel, right? It's probably not the case. So this is what we think happens at a very, like a voxel scale. But in practice, it's not hard to imagine that all these pathways, they're actually crossing each other and bending and splaying and, all these different pathways connecting each other. But so this is not a butterfly, by the way. This is a polarized light imaging microscopy image. It's an ex vivo technique, so it's not really, one of our competitors to look at brain architecture. This is a kind of a microsection near the medulla oblongata. Okay, so in the area of the brainstem. And kind of roughly, it's only this big, very small. These are the voxels with diffusion MRI overlaid on it. So the white squares, they represent the resolution that we have with diffusion MRI. So it's really coarse. It's like building blocks, right? And again, so here the color represents orientation. And you can now appreciate how complicated this network of fiber pathways in the brain is, right? So within a voxel, you've got many pathways going many different orientations. And so conceptually, you can already appreciate that an ellipsoid is never going to catch the richness of the multiple orientations present within a voxel with the resolution that we acquire with diffusion MRI. So what can we do about these crossing fibers within a voxel? Well, a lot, okay? So there's been a lot of research going on, okay? We knew this already like 2 decades ago that we had a challenge, right? And so the issue basically was that within a voxel, with diffusion tensor MRI, with this model, we could only capture one dominant population, one orientation. And I'll skip all the technical details, but with a technique called spherical deconvolution, we can now capture a fiber orientation distribution. So in any orientation, we can capture the fraction of pathways going along that orientation. So the cool thing is now, if we look at this middle area, and let me just flick between the 2 images, here you see only one orientation, green or blue. Here you can actually see multiple peaks within a voxel, showing front to back, left, right, up, down. And this is something that we know. We know that pathways interdigitate at the voxel level of 1 millimeter, 2 millimeter resolution. So how does that work with tractography then? Well, this is what you would get with fiber tractography, assuming a 1 fiber population model. And this is with multiple fiber populations. And let's just focus on a specific structure. So this is, again, part of the corpus callosum. This is a mid-coronal section, and you can appreciate pathways connecting both hemispheres. This is with the one fiber population model. This is with the multiple fiber population model. Well, obviously, if you show these images to a neurosurgeon, they'll be like, well, wow, what a difference. I mean, okay, this other model, I probably shouldn't trust it. I should choose this model, right? But again, this model also has assumptions. It might not be perfect either, right? So this is the danger of using this technology just blindly in pre-surgical planning, okay? But at least what you see here is something that we're familiar with. You see the hand knob, if you're familiar with the homunculus we've shown in the previous presentation as well. So this makes more sense. We have the collateral projections of the corpus callosum. So we assume that this model is more accurate, okay? And this is kind of our curse. There's literally hundreds of models nowadays, and everybody has their own. And like many artists, diffusion MRI scientists have the bad habit of falling in love with their models, right? So everybody's proclaiming, I am the best. No, I'm the best. And so one of the great challenges now is to come up with, a good ground truth, or like, other models, things that we can combine to have a better idea of what is correct. Something that we've worked on a while ago was related to visualization. Okay, it's one of my brainchilds, something I love doing, playing around with visualization techniques. And when you're looking at this kind of whole brain tracking image, it can be quite cluttering. You would like to see deeper into the image. And so what we came up with was something very trivial. And if you have a line, and that line is pointing towards you, right? Like in this image, all the blue lines, right, going up, down, so you're looking from the top. Ideally, you want to get rid of that line because it becomes, ill-defined in a way. Is it a point? Is it a line, right? If the line is in your axis, you want to get rid of it. So what we came up with was an orientation-dependent rendering of transparency of the lines. And you can do this in many ways, but if I show the same image, but then getting rid of all the blue lines in a rendering opacity kind of way, you get an image like this. And this has actually led us to the definition, or you could call it exploration, of a new pathway in the brain, the supero-anterior fasciculus. So it also feels like neuroanatomy is in a new era. Like we get this new technology, and we find new pathways that we couldn't see before. I mean, we had Klinger dissection, we've got neuroanatomists doing their dissections, but that's also an art in a way, because you're peeling off the white matter. I don't know if anybody of you has ever opened the brain and, played around with it. Probably not, hopefully not, unless you're a medical student, okay? Sorry, bad thoughts. Anyway, if you're doing this is also an art, right? And in some way, what we're doing is also an art, but looking at this new visualization, something immediately pops up like this structure, right? Something we didn't see before, partly because it was all cluttering each other. We couldn't see it. And then looking back, so this was basically showcasing every pathway left-right transparent. I could also say, let's only keep those pathways that have something going up-down as the dominant orientation. Okay? And again, you could see this kind of pathway popping up. And if you look at the conventional diffusion color-encoded maps, you could already see like the blue dots here. So that would be like over here, this cross-section here. And here it would be the dominant population within a voxel showcasing the blue orientation here. So here you see some more details. If you look at this map, there seems to be like this discontinuity between the blue and the green. But this was the case with the old model, right? Remember, you only have one orientation. But with the new model, you can clearly see that there's this multiple pathways crossing each other. So something is going up, down, something is going front, back. So with these new models, we were able to reconstruct this pathway. So the question still remains like, is this a genuine pathway? And obviously we did a lot of verification afterwards, and I'll come back to that in a moment. The first concern was, this is just a singulum bundle. And we were like, well, no, here's the singulum bundle in red. This is the other pathway. Okay, it's something else. And then we did the dissection, right? And then it's a bit, self-fulfilling prophecy, wishful thinking, whatever. But you could see this pathway here in light blue. By the way, in red, this is the singulum bundle again. And then we did like another technique, what I showed you earlier, polarized light imaging microscopy. And there was also suggestion of pathways connecting. So again, this is kind of in its infancy as well. But what I try to do, in general in science, right? You have an idea or you have an observation. What you should try to do is diss it. Try to find a way to negate what you believe. And if you can't do that with many, many, many tools, then it might be true, right? So we found this pathway and we said, it's probably an artifact, it's a model or whatever. And then we tried different acquisitions, different centers, different people. We took all the variability in the acquisition and the population. Oh, it's still there, okay. Okay, let's use the standards from, Klinger dissection or regular dissection. And then we asked the neuroanatomists, can you disprove this? Oh, no, it seems like it could be. They always say, no, it could be there. And it's the same with PLI, polarized light imaging. Yeah, well, it could be there. So the conclusion now is, the SAF could be a genuine pathway. And interestingly, if you look at what regions it connects, some neurosurgeons actually told us we see some deficits in some of our operations that could be explained by cutting this pathway that we didn't know that existed. So from a functional point of view, that also made sense. And finally, I want to touch upon some latest developments. Connectomics, similar in analogy with genomics. Connectomics is like the study of the network of connections. We don't look at the brain anymore like different regions, but at an integrated network of connections. And I've been mainly discussing the brain, but any tissue which shows this diffusion anisotropy can be imaged. So also peripheral nerves. So in this case, the lumbar and the sacral plexus, for instance. Okay? So this is actually an ex vivo case. You could even see the nerve endings of the roots going into the spinal cord. This was, I think it was a crime case. So who knows, maybe CSI Miami, some diffusion tensor MRI in the future. Who knows? And then finally, anybody any idea what this is? Well, obviously it's really difficult. Well, it's a tractography result. But this is actually part of the kidney. So urologists are now also very interested in this technology. Looking at the medullae of the kidney, how these pyramidal structures also can be imaged. Right? Also muscle tissue, looking at muscle architecture is becoming important, increasingly looked for. The heart poses some challenges with the acquisition because it beats, it moves, but we can also do that. By the way, because this is a kind of an educational, like a summer school, here's a background book. We call it the Bible of Diffusion MRI, so feel free to have a look at that. If you don't buy into all the scientific and clinical aspects, there's also other applications such as 3D printing, right? So yeah, nowadays you can even print in gold, so it gives you very nice gifts if you want. Obviously, this is not a one-man show. I'm very grateful to the team of PhD students and postdocs working in my lab. And finally, I would like to thank you for your attention.